everyone joining us online can hear me. I think you can. So welcome to our first summer seminar of the year. Very exciting. We're now in person again, the first time since COVID. Very exciting. So for anyone new, my name is Chad Simmons. I'm an ecologist here at the Mersey Tobiatic Research Institute. But before we begin, I'd like to acknowledge that MTRI is in, and we do most of our projects in the Gascoic Priority Place and homeland to the Mi'kmaq people. And we want to thank the Mi'kmaq for sharing their homeland with us, and we acknowledge the treaties of peace and friendship. So anyone new to MTRI, we are a research-based nonprofit, and we're nestled in the heart of Southwest Nova Scotia, just down the road from Ketchimakujik National Park and National Historic Site. And our mission is to promote, conserve, and sustain biodiversity in Southwest Nova Scotia, or Gascoic, as well as beyond. So tonight, I'm very pleased to welcome Taryn Muldoon. Taryn is finishing up her honors project at Acadia University on bats and white nose syndrome. So very excited. And just a reminder for everyone online, please keep your mics muted. And we do have a question period at the end, or you can type them in the chat. We're also on Facebook. I'll read your questions out loud to Taryn. And yeah, Taryn, over to you.
Good. Hi, everyone. Thank you so much for attending. I'm really excited to tell you guys about bats and the research I've done here in Nova Scotia. So just a little bit of background about me. I'm an Acadia student. This was my honors research, and now I'm continuing for another summer. I started working on working on bats in Scotland when I used to work as a wildlife rehabber. I ended up rehabbing a ton of bats, fell in love, and came to Acadia where nobody studies bats. And two of my professors saw my passion and helped make this project happen. So really incredible supportive community, and this research that I've been doing has been in collaboration with MTRI, and I've received a lot of mentorship here. So it's just fantastic. So let's get into this. So I wanted to talk a minute about bats. They are incredibly diverse. These are just a couple of the very cute faces of some of the many species that exist. There's actually so many species of bats in the world that they're 20% of all mammal species. So if you put all the mammal species in the world in a hat and pulled out 5, you would pull out at least one bat. So incredible. And we're very lucky in Nova Scotia. We have 6. So one of the reasons why bats are really important is that they're a keystone species. And a keystone species is a species where if you take it out of the ecosystem will not be the same. So if you took the redwoods out of California, so incredibly important to the ecosystems that they're a part of due to their insect control, and some species of bats are seed spreaders or pollinators. But out here, a lot of what they do has to do with regulating insect populations. They're also very vulnerable, which makes them an indicator species, which is a species where if something goes wrong in the ecosystem, they're the first ones to be affected. And so indicator species we can watch to see if something is going wrong, and they let us know. And of course, something did go wrong with bats. And that is white nose syndrome, which I'm sure everybody has probably heard of, but we're gonna talk about this for a bit, because this is a big deal. White nose syndrome was introduced to North America in 2006. It started in New York State, and it's a fungus that appears to have been probably brought over from Europe on beaver uniforms or something, people not washing their equipment. And bats in North America had no protections against it, and it devastated them. I read a paper one time that called it the greatest wildlife tragedy of North America, because it has killed out like 95% of bat species of bats. And the ones that remain are in very low numbers in many places still. And how it does this is the fungus grows in caves at very low temperatures, and it will colonize the wings of the bats. It'll start growing on the bats because their body temperatures are very low when they're hibernating. So the bat's asleep, the fungus starts to grow on the bat, it interrupts many processes of the bat, and the bat starts to lose a lot of water, and it also starts to lose a lot of fat. And it tries to wake up, heat itself out, and eventually they don't make it through the winter, they lose all their body fat and starve to death. And so that's called, so white nose syndrome is caused by this fungus, which I'll be calling PD, because Pseudogenomyscus destructans is a bit of a mouthful. So this is a map. It's not the most up-to-date map, but I just wanted to give you guys an example, sort of an idea of how white nose syndrome is spreading, because it is currently still spreading across North America. It started at this red dot here, and it has not fully reached the West Coast. So there's some areas where bats are starting to try to recover. There's some areas where bats are still in a phase of dying off, and then there's other areas where white nose syndrome hasn't been hit yet. And so biologists are frantically trying to get all the information they can to protect their bats before it gets there. And so the ways that we gather information about PD, there's multiple. Testing for the fungus that causes white nose syndrome is often done by swabbing the surface with a swab that's like a Q-tip. So you can swab the actual bat itself to see if the bat has any spores on it, or you can swab the environment the bat lives in, like this bat box here, or you can collect the bat's poop and you can test the poop for traces of DNA of the fungus. And that DNA would be in the poop if the bat had any spores, if it's fungus on its fur, and it would be licking it off, ingesting it, and the DNA would still be coming out in the poop. Which is what I did. Spoiler. So this figure is from a study in the States showing the survival statistics in the tricolor bat, which is this little cutie here. And so you can see it starts with a tremendous die-off when white nose syndrome hit this particular cave that continued over multiple years. And then slowly populations started to rebuild in the past few years. And this is a pretty common trend in a lot of places in the States. A lot of areas are starting to see slight signs of recovery. But that is not everywhere. And we're not sure if that's the story of Nova Scotia yet. So Nova Scotia, white nose syndrome was detected in Nova Scotia in the winter of 2010, 2011. And PD, the fungus that causes white nose syndrome, has been detected almost everywhere except for these 5 counties in white. And that's likely due to just a lack of data. It's assumed to be everywhere. But bats with PD spores or just PD in the environment has not been detected in those areas. So one of the things I was looking at in my research was, can I detect PD in some of these areas? Fill out this map a little bit. So just to give a bit of background of bat conservation in Nova Scotia. Start with a place that you might be familiar with. So for those of you who don't know, MTRI has a wonderful bat monitoring program. This year is 10 years of their bat reporting hotline, where you can call in if you see a bat. And this allows researchers to find out where bats are still being seen, how many are being seen, and get just sort of a sense of what's going on, how many bats are being seen each year, which is a really excellent monitoring program. They also have a monitoring program on maternity colonies, which are where mothers will gather in large groups to raise their offspring. So whenever MTRI finds out of a maternity colony, they will count those bats every year and see what the numbers are doing. And so for my study, I was really lucky in order to be able to work closely with MTRI and collect poop from bats at these maternity colonies that already had years of data. So really wonderful. There is also a Canada-wide bat box project study that is ongoing in Nova Scotia. So this is conducted by a researcher called Karen Vanderwolf, and she's trying to find out what is the best bat box? What do bats need as a bat box habitat? And part of that is also she is swabbing bat boxes to test for the fungus that causes white nose syndrome. And so she has quite a few sites here in Nova Scotia. And so we've been helping her with that research. And it's also some of my sites are her sites. And this is a really excellent study. If you have a bat box on your property, I highly recommend signing up to be a part of it, because she's always looking for more boxes. And that comes to me. So I was very lucky to come to Nova Scotia and be very excited about bats and have this amazing research already happening. And been talking with my supervisors, been talking with Lori Finney from the Mercy Tobiatic Research Institute. We hit upon this, which is quite the title, and I'm not going to be using most of these words in the talk, but this is my thesis. And essentially, I collected poop to find out when and where I could detect the fungus that causes white nose syndrome. And there's also a little bit to do with parasites, which we'll be talking about at the end of this presentation. So, a little bit of background before we get into collecting poop. Like I said earlier, there's 6 species of bats in Nova Scotia. There's these 3 gorgeous migratory species and 3 cave hibernating species. And these are the ones that are affected by white nose syndrome. There's a little tricolor that we saw earlier. So the bat in particular that I focused on is one that really likes to roost in human-made structures. And this is a really awesome bat to study. It's a little brown myotis, because it'll be in a bat box or somebody's house or their barn, and then you can just drive right up to their property and jump out, and the bats are right there. Your study site's right there. You don't have to go tromping through the woods looking for one particular tree. It's really great. And they are just the cutest bats ever. But unfortunately, like all bats, they are in Canada, they are endangered. They've gone through 93% population decline in Nova Scotia. I should have said not all bats in Canada, all bats in Nova Scotia, they are endangered. And that's because of white nose syndrome. So in the summer, female bats, female little brown myotis will roost in maternity colonies in habitats such as these. And of course, we know in the winter they hibernate in caves. So the big question for researchers has been, do we have to worry about white nose syndrome when they're in these summer structures? And the consensus seems to be no. But we'll be diving into that a little bit more, because a lot of my research is about when can we detect white nose syndrome. So we know that white nose syndrome kills in winter. And at other times of the year, it's not killing bats. The fungus persists in caves year-round, but it can only infect and kill bats when they are at very low temperatures when they're hibernating. And so in the winter, that's when bats will be affected. And then they leave their hibernacula, but they're still carrying lots of spores from hibernation with them. And so that was the big question. When bats get to their maternity colonies or the roosting in forests that males do, are they then contaminating that environment? And research seems to suggest that the spores can last a little while in the environment, but not really long enough, and they're not going to affect the bats anyway, because the bat's body temperature is high enough that they're not going to get sick. So there may be some spread of PD to the summer sites, but there's not going to be spread of white nose syndrome in the summer. And one interesting part of bat behavior is in the fall, they leave their summer colonies and they start to fly to caves, and they will mingle there. They'll go into the cave, they'll mate, so they'll be touching each other, they'll be touching the cave, and this happens pre-hibernation. So they're not just going to the cave and then going to sleep. There's quite a bit of mixing and mingling, and that's some percentage of bats, small percentage, but some do go to multiple caves. And so when we're looking at the life cycle of this fungus, it's affecting bats while they're asleep. And some bats may wake up and decide, screw this, I'm uncomfortable in here, I'm sick, I'm gonna fly to another cave. And then they do that. But if the other cave is far away, how is this fungus traveling from cave to cave? And so that was one of the things I was curious about. When can we detect PD on bats in Nova Scotia? Will we be seeing it in the summer? Will we be seeing it in the fall when they're swarming? And this can give us some idea of when they might be spreading it. So, as I mentioned, studies have found that bats can test positive for PD in the summer. So we know that there is some chance that they're carrying it around in the summer. PD is detectable in bat guano for quite a while, so it's a great substrate for detecting it. But yeah, what about transmission? So talking about that. But first, of course, I was working with bats that have a disease that is killing them. Had to be very careful not to be spreading that disease. So got to wear these fun suits, also the masks against COVID, rabies shot, and all the permits necessary. So everything was up to code. And these were my questions, essentially. So where in Nova Scotia can I detect PD? Will it be in every site? Will it be in some clumps of Nova Scotia? Will I be able to detect it in those counties where it's not been detected before? And when can we detect it? Will we see it in the spring when they're coming out of hibernation? Will we see it throughout the summer? Will we see it in the fall when they're swarming? And this can tell us about possibly when this disease might be being spread. So, and of course, understanding when a disease is being spread tells us a lot about conservation, because we have a disease spreading through a population. We need to know when it's being spread and when the highest risk is and how it's being spread in order to figure out what to do next. So, oops, daisy. So I collected the guano, and I did this by going to these maternity colonies and looking for evidence of bats, and checked a lot of sites for bats. And it was really exciting when we got some bats. And often we just saw poop, which is also pretty great, because the poop was what I was there for. And then I would put something down for bats to poop on, like a little bat toilet underneath where they lived. And every 2 weeks, I would come back, and the bats would have used their toilet, and I would clean up the poop and shake off the sheet and rinse and repeat all summer long. And then those samples were sent away to a lab to be tested for PD. And this is what I found. No, this is not what I found. Next slide will be what I found. So this is the distribution of sites where I did this. You can see it's fairly concentrated towards southwest Nova Scotia. We did try to find some sites to fill this middle gap, but it was difficult. But we got a fairly good range. This is not all the sites in my study, but it's the major ones. And so we found in May, almost every single site was positive for PD. So across that entire map, there was only one site that was inconclusive, because it had a very small kind of moldy sample from it. So can't tell either way whether that site had it. Every other one was positive. And then throughout the summer, it dropped. We never fully had a time where all the sites were negative or inconclusive. There were some sites there was one site that stayed positive all summer. But in general, they all became negative or inconclusive, and then sharply shot up again in September, which is when we would expect bats to be swarming. So before I get into that, I did some stats to make sure there's nothing else that was interfering with these results. So one thing I was concerned about was the age of guano, because I told you guys that I went out every 2 weeks, and that's a bit of a lie, depending on the location of the site. Some sites, like that one in Cape Breton, did not get to that one every 2 weeks. So some of the guano was older than other guano, and that means that guano has been sitting out in the elements, and it's been rained on, it's got the sun on it. What is that doing to the DNA? That DNA would likely be degrading faster than guano that's sitting in somebody's farm. But we did statistical tests, not significant. So the age of guano is not an issue here. Other thing I was worried about was some sites in the barn, some sites outside. Those ones are going to be getting a lot more degraded. Guano is breaking down faster, DNA is breaking down faster. No, wasn't an issue. So those two factors did not mess with my results at all, which is very nice. So my findings support the hypothesis that PD is being spread, at least partially through swarming. What was interesting in this study is that I was testing guano at maternity colonies. So those are not necessarily near a cave. In fact, a lot of the sites are hundreds of kilometers away from the nearest known cave. And there's very likely a closer cave that we don't know about. But so we're testing guano of bats that are not currently swarming, and it's coming back positive for PD after having previously been negative or inconclusive. So this suggests that they are somehow picking up on more spores and that is being moved across the landscape, even to bats who possibly didn't swarm or have decided to swarm and go somewhere else, like back to their maternity roosts. So that is a very good sign, or not a very good sign, but that is a sign that that this could be a driver of how PD is being transmitted. And taking a little hitchhikers and just possibly getting them to different caves. So to go over these results again, we had the uniform PD across Nova Scotia. I did manage to detect it in 3 counties where it's not been detected before. And I'm working on another one this year. And so there's potential that PD is being dispersed during spawning, that this is a high time when it could be being spread in different caves. So next, this year, I'm doing an extra test at some of my sites because I'm using the same sheet all summer, last year and this year, and same sheet between years, but all summer I've been using the same sheet. And so at some sites I've been doing tests just to make sure that the sheet isn't becoming contaminated. And so we'll see what the results of that are. I'm also collaborating with Karen Vanderwolf's Fat Box project and comparing her findings, swapping boxes with my findings, checking guano, and hoping to publish these results. Before we finish, let's talk about parasites. Because that was the other part of this research that it's a little bit shafted sometimes because I find PD so interesting. But parasites are too, because we don't know a lot about that endoparasites. So a lot of wild animals have some sort of intestinal parasites. Bats, a ton of flukes. I don't know why, but flukes are the main parasite they get. And we know what parasites they get, but we don't know a lot about which bats are getting them. A parasite maybe has been discovered in Indiana in a bat, but we don't know if that same parasite would be in Nova Scotia. In fact, we don't know at all what parasites are in Nova Scotia because nobody's ever formally tested it. So I had all this poop and I had the DNA from the poop. And so we've been trying to develop primers, which is primers can sort of tell you what you do it right. If a particular parasite is in the poop that you're looking at. And so we'll look to see if we can find any parasites. I also had a dead bat that a member of the public found, and it was very fresh. And a fresh dead bat is pretty hopeful that we might have some parasites in that. So I took it to the University of PEI, it was dissected there, and a very good parasitologist took a look at the parasites. And we found this parasite, which is called Urochunus gabardum. That is a beautiful picture of that parasite after we stained it. And this parasite, we did know about this parasite in bats, but it's not been, I can't find any literature mentioning it being detected in Canada before. So this is really cool. And when we tested some sites with some primers, just kind of seeing like, do we have parasite DNA in these samples? And primers said for some of the sites, yes. So we have some bat parasites, and we're working on more specific primers now to be more like, we have like this parasite. And for that, I'm collaborating with a wonderful friend of mine and who is working with me on the research this year. Ricky is also in the room right now. And Ricky is excellent at lab work and very passionate about DNA work. And so I'm very excited to sort of crack this case more with her and get deeper into parasites. And so just to wrap it up, I wanted to talk about what you can do to help bats. So I already talked about MTRI's bat hotline, and I have a slide at the end that has the hotline number on it. If you see a bat, report it. If you have a colony of bats, please report it. You can also put up a bat box, particularly if you have bats in your house and you don't want them and you're trying to get rid of them. Putting up a bat box gives them somewhere to move into in that process. But it's also a good habitat if you have a bat that's passing through that wants somewhere to sleep for the night. And then you can do a bat box study and give more data to Karen. Probably the best thing you can do though is encouraging and protecting habitat for bats. So having pollinator gardens, encouraging insects, preserving wetlands and old growth forests is incredibly important. And so if you have those features on your property, or if there's some nearby that need protecting, that's an excellent way to help bats. There's also been a lot of research that a lot of people have found that bats really like patio umbrellas and awnings. And so this becomes a bit of a problem in camping season, like right now, because bats will person somebody's awning for the night, and the next morning, the person doesn't realize they have a hitchhiker and they'll drive off, and this bat is suddenly just whisked away from its habitat and taken somewhere completely different, and that's not very good for the bat. So if you're camping and you have an umbrella or an awning, check it before you leave, just to make sure you don't have a little friend with you. And this little baby flying fox says, "Here you learn, break the stigma." And I'm going to give some thank yous. So thank you to my funders, the Women and Activist Society and NSERC, and of course MTRI, which this research has been in very close collaboration, and I'm so grateful for having me here today and every day. And after this, I'm actually going out to collect more guano, so it continues. And a big thank you to research assistants and particularly landowners. I don't know if any of you are on this call right now, but they let me put a sheep in their yard all summer and come every 2 weeks to collect poop, and that's just incredible. So thank you all very much. Have a good night.
Thank you, Taryn. Very well done. If anyone has any questions, you can unmute yourself if you're on Zoom, and if you're on Facebook, you can type in the chat, and if you're in person, you can talk.
I have a question. It's more about covering. How exactly are they covering if this is still out there?
So there's a few different ways that they could be. There could be behavioral changes that they're making, or there could be genetic changes. And
Because obviously you're
yes, it's interesting. I've read some papers that said that myotis species, or jewel ground myotis, is a myotis species, and several others of bats that are really affected. In Europe, actually still do get spike syndrome sometimes and have die-offs, but not as strong.
No.
So it seems like myotis across the map are more susceptible. But the strongest evidence that I found for recovery, just from reading the research, is that it appears to be their wing microbiome that could be potentially be playing a role. That there's certainly parts of the microbiome that are more protected. And they found that big brown bats are not nearly as effective as myotis species. And there's been some people hypothesized that it's like the body size that we can just survive longer because they're fatter. But then some people looked at that wings and discovered that big brown bats are actually just getting less sick and fewer wing lesions, less growth. And they realized that it could be something to do with the microbiome of big brown bat wings, that some elements that the little browns are missing. So, yeah.
Sandra asked, could you please repeat the name of the lady regarding the bat box? I think that's Dr. Taryn.
Yeah, Taryn Muldoon. Should have written it on the slides. You don't have to spell it, you can put it in the chat.
I don't know. You can say Google Canada Bat Box study.
Oh, yeah, it's
Might be easier.
I can go back to the slide if people want to take note of the slide. There. Any other questions? Let's screenshot on that page.
Oh, that's it. Great. Thank you again, Taryn. Excellent job. And thank you everyone for joining us. As I said before, with our summer seminar series, you can join us in person in Kempt, just down the road from Kejimkujik, for any of our summer seminars, which we are doing weekly, every Thursday, 7 to 8 p.m. And we have snacks. So with that, thank you, everyone. I'd like to thank the Region of Queens for supporting us and making all of our seminars throughout the year possible. Great. Thank you, everyone.
Thanks. That was great, Taryn.
Yeah.
Thanks, Taryn.
That's Jane.
She's like, I'm gonna put my husband there.
Oh, we have one more question. So Sarah asks, in your opinion, what knock-on effects are there on the environment without these key ecosystem species?
You, Sarah. So does she mean here in Nova Scotia, or a lot of landlords, a lot of landowners have told me that when the bats disappeared, their bugs increased on their properties. And I can't say I'm an expert on knock-on effects of the disappearance of bats. But if you have more bugs or a specific species of bug, for instance, particularly if it is a bug that spreads some sort of disease or is itself a parasitic bug, a lot of moths cause a lot of damage to crops. So if you were to take bats, I don't know if I can remember this off the top of my head, but if all bats were to disappear out of North America, there would be a $1.3 billion loss in agriculture due to all the crop pests that would then proliferate and all the money that would have to be spent on pesticides. So from a human economic perspective, they have a huge control of crop pests, but they also would be controlling a lot of other insects in the ecosystem. You know, maybe certainly some blood-eating insects, but also a lot of moths that might be causing damage to other plants that we don't care about as much. And then if you consider the other services that bats provide in other areas, they're key pollinators for many species of plants, including some cacti that they pollinate, and they're also really important seed spreaders. So if you were to take bats out in those areas, you'd have some plants that would be unable to be pollinated, or would not have their seeds spread, and some forests not be regenerating as well.
Great. Okay. Thank you, everyone, once again. Hopefully we'll see you next week. Cool.